Today, we're diving into something truly mind-bending. We're looking at how artificial intelligence is starting to read and maybe even write the very code of life itself.
Okay, so where does this all begin? Well, it starts with a flood, an absolute explosion of data. You see, thanks to incredible technologies like high-throughput sequencing, we are mapping out the biological world, from our own genomes to the tiny proteins that make our bodies work, at a pace that is just staggering.
And that right there is the central problem. It's one thing to have all this data, right? But to actually make sense of it, to understand its insane scale and complexity, that is a whole different ballgame. It's like having a library with billions of books written in a language we're only just starting to figure out.
To put it bluntly, biology is facing a data tsunami. And our first job here is to just get our heads around the sheer scale of this information overload. And listen, we're not just talking about one type of data. No, this deluge is everything. It's the genomic sequences, which are a fundamental DNA blueprint. It's transcriptomics, which is like reading the active messages being sent inside our cells. Then you've got proteomics, which looks at the actual little machines of life, the proteins. And now, we can even get these incredibly detailed snapshots of single individual cells. It's a lot.
So, how in the world do you navigate a tsunami like that? Well, the answer, according to this research, is a powerful new tool. You should think of AI as our new microscope, but one that lets us see the hidden patterns swimming in this vast ocean of data. Now, AI isn't some single magic bullet. It's more like a powerful toolkit. Researchers are basically using three main approaches here. You've got your traditional machine learning, the much more complex deep learning, and this really interesting trial and error method called reinforcement learning. So let's just quickly break down what each of those means.
So how are they different? Well, think of traditional machine learning like a really good detective who's great at following clear, well-defined clues. Deep learning, on the other hand, is like giving that detective a supercomputer that can find these super subtle, complex connections in a literal mountain of raw evidence, like an entire genome sequence.
And then there's reinforcement learning. This one's the pioneer of the group because it learns by doing. It's like an AI chemist in a virtual lab, trying out millions of different combinations to design the perfect drug molecule, and it gets a little reward every time it gets closer to a good result.
Okay, so this technology is amazing, but what does it actually do for us? Let's take a look at how we get from analyzing biological code to actually developing real-world cures.
And the source paper is really clear on this point. The results we're seeing, they aren't just small little steps forward. This is a fundamental, revolutionary shift in how we approach biology. And what's really wild about this is the sheer scale of the leap we're talking about. These are performance improvements of not 10, not 50, but in some cases, well over 100 times better than traditional methods. I mean, that's like jumping from the Stone Age to the Space Age in just a few years.
And this is the perfect example. For 50 years, predicting how a protein folds up into its complex 3D shape was like the holy grail of biology. And then AlphaFold 2 came along and basically cracked it with near atomic accuracy. That tiny number, 0.96 angstroms, that represents the difference between just guessing and actually knowing the shape of life's most essential machines.
And it's not just about the shapes. It's about understanding the language of proteins. This data shows that modern AI can figure out what a protein does based on its structure 135 times more effectively than older methods could. This just unlocks a completely new level of biological understanding.
But it's not all smooth sailing, of course not. To really unlock AI's full potential, there are still some pretty big challenges we need to figure out.
For one, the data itself can be noisy, messy, and incomplete. Our genomes are incredibly long, which creates huge problems for computers. And this one is critical. We need to understand why these AI models make the decisions they do. We have to crack open that black box. And all of this has to happen while making sure patient data is handled ethically and kept private.
And here's a major reality check from the research. The incredible power of these new models comes at a really steep price. You see, training a traditional model might take a few hours on a normal computer, but these huge new foundation models, they need massive clusters of specialized computers running for weeks or even months straight.
So, where do we go from here? Well, the paper argues pretty convincingly that the path forward isn't one that any single group can walk alone. It has to be a shared journey.
This visual from the source really sums up where we are right now. It's a balancing act. On one side of the scale, you have these enormous challenges. But on the other, the opportunities in medicine and research are, well, they're truly unprecedented.
And the paper's most crucial takeaway is this. To make that next big leap, this can't happen in separate silos. Real progress depends on building an open, collaborative ecosystem where biologists, computer scientists, and doctors are all working together.
So, for the first time in history, we have a tool that can read the book of life at a scale and speed we never even imagined was possible. The question is no longer just can we read it? The real question now is, what new chapters, what new cures, what new discoveries, what new understanding will we ask it to help us write?